All right, here we go then. So this is Science Saturdays, and everything that I have presented up till now brought us to today, which is the one that I have been most excited about, and that is to talk about viruses and vaccines. All right, so just as a little review from a couple of previous presentations, the types of infectious agents that are most relevant to human disease are categorized as acellular, prokaryotic, or eukaryotic. Today's focus is going to be on our acellular pathogens, specifically viruses. So these viruses had been discovered in 1892, so well over 100 years ago. The first virus being tobacco mosaic virus. And then the whole field of microbiology and infectious disease is actually well over 200 years old. And so viruses were first discovered in plants. There is no great conspiracy. Viruses exist. Bacteria exist. So to learn more about infections, I would like to direct you to the January 17th presentation of Science Saturdays, which focused on infectious disease. So just as a little bit more review, viruses are small. Viruses are imaged using electron microscopy. Here is an example of what that ends up looking like. This is our coronavirus, which was named for this corona like appearance. This is the spike protein that is on the outer surface of each one of these virus particles. So viruses are obligate intracellular parasites. Now, what does that mean? That means that viruses must live and reproduce inside a living host cell. So all parts of their replication techniques take place within a living host cell. And this is because viruses are small. They lack the necessary machinery to survive outside of a host cell or replicate independently. So they take advantage of host cell machinery to they themselves replicate and spread. Their entire life cycle is intracellular, meaning that although they exist on the outside of cells, their actual life cycle is intracellular. And just to review, this is the central dogma of molecular biology. So what this is we are all made up of DNA, so deoxyribonucleic acid. That is our genetic code. Our DNA is replicated through DNA polymerase. It is transcribed into messages, and this message is known as that RNA, and that's done through RNA polymerase. And then this message is translated into the actual functional output, which is a protein. So the DNA is double-stranded. It's composed of 4 nucleotides, adenosine, thymine, guanine, and cytosine. It is also known as our genetic code and is stable. And this is important to remember because when it becomes unstable, we have things come up like cancer. So DNA polymerase in and of itself is a high fidelity enzyme, so that replication is happening with proofreading taking place because you don't want to introduce things like mutations. And the DNA is not degraded, meaning it is stable. And contrast that with RNA. RNA is a single-stranded molecule in humans, and it is composed also of 4 nucleic acids, adenosine, uracil, guanine, and cytosine. And with respect to today's conversation, we are going to focus on one specific type of RNA called mRNA or messenger RNA. And these are the messages from our genetic code that are then translated into proteins. And RNA molecules themselves are relatively unstable. And that is important because that is what influences things like protein expression. And so if you think about it in respect to immune cells, you don't always want your immune cells on or off because that's not their job. And so the things that kind of turn these immune cells on and off is the expression patterns of different proteins, which in and of themselves are dictated by the expression patterns of mRNA. All right, so now that we know or have reviewed that central dogma, viruses can be DNA viruses or RNA viruses. Further, they can be composed of single-stranded DNA or RNA and double-stranded DNA and RNA. An additional level of complexity is that some viruses use reverse transcriptase for host integration, and some of these viruses also come with RNA-dependent RNA polymerase, or RDRP, packaged within those viral particles. And this will make a little bit more sense right here, where we review the central dogma of biology and how viruses fit in there. And so what we have is, once again, our DNA here as the double helix in blue. We have the single strand of RNA. In this case, it's identified as positive sense or negative sense. Positive sense RNA is the type of RNA that can be directly translated into protein. Negative sense needs to first be made into a positive sense before it can be translated into proteins. And so that reverse transcriptase belongs right here, where we have an RNA that can go through and create a intermediate product and then have this incorporation into the host DNA. And RNA-dependent RNA polymerase belongs right here with this whole conversion of negative, or not conversion, but the transcription of negative sense RNA into positive sense. And so the takeaway here is that the viruses are really complicated despite being quite simple in nature. And so they're incredibly small. They don't code for a lot of things, but they have this incredible amount of complexity, and that impacts their infection and their general viral life cycle. So, one of my favorite ways of looking at the different classifications of viruses is the Baltimore classification. And this was developed back in 1971 by virologist David Baltimore. And it has since been amended with the discovery and classification of the class 7 viruses, which are double-stranded DNA with reverse transcriptase. So this is what that looks like. So we have 7 groups of viruses. And this classification basically identifies what these viruses are made up of and what is needed in order to get to that messenger RNA. So, and that's important because we go from that messenger RNA to those functional proteins, and those functional proteins are required in order for viral spread. So first we have our group 1 viruses, and these are double-stranded DNA viruses. They're very exciting. These are some of my favorites. They include viruses like human papillomavirus or HPV, the adenoviruses, which can cause infection in a whole bunch of different ways. A big one being respiratory, so up in the upper respiratory system as well as the GI system. We also have the herpes viridae class of viruses, which includes herpes simplex virus, Epstein-Barr virus, which can create or lead to infectious mononucleosis, varicella zoster, which is known as chickenpox or shingles. We've got cytomegalovirus or CMV, which can also present with infectious mononucleosis. And then there's this HHV6, and this is a really interesting herpes virus that plays a role in a lot of different types of human disease. And I've bolded a couple of these, HPV, EBV, and HHV6, because these are really unique in that they're double-stranded DNA that can actually integrate into host DNA and have been looped into some human conditions like cancer. And another interesting thing with these viruses in particular is that they can persist in the latent stage, meaning that they can avoid immune surveillance. And so these viruses can sit quietly inactive within these cells and wait for an opportune moment to reactivate. So this might be times of stress, or it could be a different infection. And so that is especially important with like varicella zoster, where we have a person that comes down with chickenpox when they are young and ends up with shingles when they are older. That is a really great example of that. And then moving on, we've got our group 2 viruses. And so these are single-stranded DNA viruses. One of the big ones that are important to human health is parvovirus B19. And this leads to the condition of fifth disease. And something that's really interesting with fifth disease is that these were named based on the five most prevalent causes of childhood rashes. The first being measles, the second being scarlet fever, then rubella or German measles, and then it was fourth disease or Duke's disease, and then the fifth was parvovirus B19. And it's important to note that the second, scarlet fever, and the fourth, Duke's disease, are both bacterial in nature, but measles, rubella, and parvovirus are all viral. And then we move on to class three. These are double-stranded RNA. And the really big one is rotavirus. And so rotavirus causes GI distress, especially in children. And then we have the class four, which are positive sense single-stranded RNA viruses. And these include things like SARS-CoV-2, which is also known as COVID-19. And then we have two of the hepatitises, hepatitis A and hepatitis C, as well as rhinovirus, which is known as our common cold. Then we have the class five viruses, and these are negative sense single-stranded RNA viruses. And this includes things like influenza, measles, mumps, RSV or respiratory syncytial virus, and rabies. Then we have class six, and these are positive sense single-stranded RNA with RT, so that's reverse transcriptase. And the two really big ones here is HIV, human immunodeficiency virus, and the HERVs, which are human endogenous retroviruses. And these HERVs in particular are really interesting because these are ancient. And so these are viruses that our ancestors contracted and passed down through the germline. And once again, these are two viruses that become integrated with the host DNA. And then our last class is the class seven, and these are double-stranded DNAs with reverse transcriptase. This one being Hep B, which has gotten a lot of publicity lately because of the anti-vaccine measures for Hep B. But this is also one that can easily integrate into the host DNA and is also linked to things like cancer. So that would be liver cancer. So mm, yeah, I can't remember exactly which one, but it is linked to a liver cancer. So I've highlighted these groups in pink because these are all DNA viruses, meaning that the DNA polymerase is high fidelity, meaning low mutation rate. Some of these can integrate with host DNA, and it will persist over the long period of time because remember, DNA is a relatively stable molecule. So these are ones that once you get it, it's really hard to clear, if ever. And then we have this little splat here. This is a lower fidelity. And double-stranded RNA is an immediate indicator for our immune system that it is foreign. We don't have double-stranded RNA within our own system, so this elicits a potent immune response. Doesn't mean that you don't get sick. You can totally get sick, but it is one that stimulates a really robust immune response. And then we have our RNA viruses down here, and these are they use lower fidelity machinery, meaning that it can result in rapid evolution or rapid evolution through mutation. And I've included the dsDNA RT class of viruses down here because these use lower fidelity machinery. And so reverse transcriptase is an error-prone viral or is an error-prone piece of viral machinery, and it actually introduces lots of mutations, very rapid evolution, which means it's really challenging for our immune system to find and eliminate. And so these can lead to chronic infections such as AIDS in the case of the HIV, and also chronic infections with Hep B, which can lead to cancer. So that was a lot of information, but basically all it was to show you that all of these viruses really truly are incredibly complex, even though they are quite simple in nature. So host evasion through mutation, the mutation rates of different viruses are impacted by polymerase, so the RNA polymerase for replication, primarily because there's little proofreading that takes place, and that's contrasting those DNA viruses. And reverse transcriptase, like I had mentioned, is quite error prone. And then viral evolution can also take place through periods of long infection or persistent infection. So if we think back to some of the most current events, COVID-19 is huge for this. It is pretty firmly established that those that have been infected over the long period of time are some of those that are driving those mutations, which leads to each successive wave of COVID-19. And the reason for this is that when that infection takes place over a long period of time, it gives ample opportunity for mutations to occur. And so, as long as your immune system isn't clearing it or um, anything like that, you have this likelihood that replication is going to happen and mutations are going to exist. And then viruses that are especially good at infection will not only exist but be spread. And then there's also this idea of recombinant infections. And it's not an idea, it's actually been observed, especially with COVID, where somebody ends up coming down with two unique strains of COVID-19. And that's where we have multiple variants that recombine. And that creates effectively a new strain. So, on that note, here are some of the major strains and the mutations that took place, and these are at the amino acid level. So, these are with respect to, in particular, the spike protein. And you can see we've got alpha, delta, and the Omicron collection of mutants. And then you can also see that the different antibodies are most effective at targeting some of these different mutations, some of which aren't very effective at targeting some of these different mutations, and then how much of the virus actually escapes immune response. And so basically, what this is showing is that mutation drives this whole recurring waves of viral infection. And then we also have uh, this process of segment mixing, and this is especially relevant to the negative sense single-stranded RNA viruses. And so this includes things like influenza, and this is the recombination of different segments of genetic material. And that is what drives some of the protein expression on the surface of these viral particles. And so when we refer to H1N1 or H3N2, we're referring to the different or the different surface proteins on those viral particles. And so this is especially important for the viral proteins that mediate cell entry. In the case of influenza, that would be the hemagglutinin or the H. And so that brings us to influenza. And so influenza's um, mutations are due to, or not mutations, but the seasonal effects of influenza are in part due to this antigenic shift and antigenic drift. So let's talk about that. Drift refers to point mutations, whereas shift refers to that recombination that we just talked about on the last slide. And also, this includes things like being multiply infected. And so that would be infected with both H1N1 and H3N2 at the same time, or maybe different subclades of the same viral class. So that might be a subclade J and subclade K of H3N2. And so those are just different unique viruses. And so within the influenza subtype. So hemagglutinin or hemagglutinin is the protein that is involved in viral entry into our human cells. There are 18 subtypes, but the ones that are most relevant to humans are 1, 2, and 3. However, we have had H5 and H7 become of particular note for humans. And then we have neuraminidase, which is involved in virion detachment. So after the virus has been replicating within a cell, neuraminidase is involved in the release of virus from those infected cells. And there are 11 subtypes for that of particular importance is 1 and 2 for the human condition. And so what this looks like is we have the H and the N. These are our or are the proteins that are expressed on the or on the surface of this viral particle. And we have a drift event. So this is mutations that then affected the hemagglutinin protein. And so what we see here is the change from a five-sided to, the star-shaped. And this is all just in theory, but it effectively affects the ability for this virus to bind to human cells. And so then we have the process of antigenic shift. And so this is where a person becomes infected with two different viruses. And within that host cell, we have mixing that takes place. And so then we have the development of a brand new viral particle. So that leads us into this conversation of vaccines, because one of the big things that I often hear from people who don't understand or were never taught any of that information about influenza is, why do we need a booster every single year? Well, remember that viruses are obligate intracellular parasites. And the idea of vaccines with respect to viral vaccines is to target the extracellular virus before it infects the cell or during that active replication process. And so a vaccine needs to be able to allow the immune system to find and eliminate infected cells and viral particles in the extracellular space. And an important thing with vaccines is that it must target viral components. And these are, of course, viral vaccines. And that's important because you don't want your immune system going after yourself. And so if we think about this, really the best proteins to target for vaccines are the proteins that are on the surface. So these might be cell surface proteins like the H3 or the N1, or it could even be some, in the case of non-enveloped viruses, it could be some of the proteins that form this capsid here. And so we have an enveloped virus over here. We have a non-enveloped virus over here. So for this, we're not going to talk about the difference in natural versus vaccine-acquired immunity outside of the fact that it stimulates the same mechanisms, but vaccines get you there without a full-fledged sickness. For more information about this, go back a couple weeks to the January 10th Science Saturdays, where we discussed the immune system. And I think something that is important to talk about, though, is that there are several different types of vaccines. And so the first is the live attenuated or vaccines, and these use weakened virus, and that includes the measles, mumps, rubella vaccine. And so this is live virus that has been attenuated in such a way to not cause a full outbreak of the disease, as well as varicella or chickenpox. And then we have the class of vaccines known as the inactivated virus vaccines. And so these use virus that has been inactivated or killed, and that includes polio and influenza. And so these viruses have been, the whole virus itself is included within the vaccine, but it has been killed in such a way that it is no longer infectious. Then there is the subunit vaccines. And so this can use polysaccharides or conjugates. So these can be proteins that are attached to something else, or protein-based vaccines. And this would be like Hib, pneumococcal. So this is like pneumonia. And then shingles for a virus, and Hep B for a virus. Then we have the toxoid. And so this targets the toxic activity of bacteria. So this would be tetanus and diphtheria. Then we have the viral vector. And so this uses a genetic code. Some of our COVID-19 are viral vectors. Some of our COVID-19 vaccines are viral vectors. And then there's also an Ebola vaccine that's coming down the pipeline. And then our last is the mRNA vaccines. And these use viral mRNA to train the body. And a big one here is COVID-19. And so a big question is, how do these mRNA vaccines work? How did they get pushed out so fast? Well, they didn't actually get pushed out all that fast, because this is something that had been investigated for several decades prior to the fast track of the COVID-19 mRNA vaccine release. And so effectively what ends up happening is that we have a nanoparticle RNA that is delivered by syringe, and those capsules fuse with the cell membrane. And then that genetic material is released. And what ends up happening is that human cell then makes the target protein and can express that protein on its cell surface. These would be our antigen presenting cells or APCs. We talked about some of these in our immune system talk on January 10th. And when that cell presents that target protein on the surface, it can actually activate the immune system to respond to that particular protein when the human system is infected. And so let's say that you get exposed to COVID-19, it can detect some of those proteins. And so, of course, this is specific to a protein. And so in the events of mutation, then all of a sudden we start losing some of that protection. So, some of the breakthroughs that have been made with these mRNA vaccines since COVID-19 are really quite incredible. And so that includes RSV. This is a vaccine that has been approved. It is recommended for all adults over the age of 70 because it disproportionately affects two populations, our seniors and also our newborns. And so adults over 70 are recommended, and then high-risk adults, so those that have chronic respiratory issues, between the ages of 60 and 74. And then also, this is new women or pregnant people between 32 and 36 weeks. And then with respect to the clinical studies that are coming down the pipeline, they have mRNA vaccines targeting cytomegalovirus or CMV, as well as influenza, shingles, Zika, HIV, and Lyme's disease, as well as in earlier stages of development. So these are some of the phase 3 and phase 2, and these are in phase 1. This includes tuberculosis, genital herpes, norovirus, and malaria. And then there's some progress being made in non-infectious disease. So the idea here is that if you can target a specific protein that is being expressed in an inappropriate way, you can target things like cystic fibrosis and other rare diseases, as well as cancer. And so some of the things that are happening at the mRNA vaccines against cancer, we've got clinical studies that are in phase 3 for melanoma, as well as various stages of clinical research for pancreatic cancer, non-small cell lung cancer, colorectal cancer, and ovarian cancer. And the way that these work could be either through personalized treatments. And so in this case, this is customized for the patient's tumor makeup. So this is really truly getting into that whole area of personalized medicine that we have been very slowly headed towards for the last couple of decades. But also, it could be more generalized where it targets specific tumor proteins. And another thing, too, is that these mRNA vaccines have been shown to kind of wake up the immune system by acting as a siren to alert the immune system. And so, in some of the retrospective studies that have taken place since the COVID-19 mRNA vaccines were released, it was shown that some people who ended up with the COVID-19 vaccine actually showed a significantly better outcomes with respect to cancer progression, especially as it pertained to melanoma. And this is a COVID-19 vaccine, not a melanoma vaccine. And so somehow these mRNA vaccines are impacting our immune systems in other ways as well that help with the surveillance of not just viruses, but also things that are going wrong in our own bodies. So with that, that leads us to our next conversation is once you get that vaccine, why do some need boosters and some don't? Well, some vaccines will lead to a lifelong immunity, and those are generally for those viruses that are a bit more stable and unlikely to mutate. But we do have some that require boosters. And so this is for tetanus, diphtheria, and pertussis, or the Tdap. And this is every 10 years. And that has to do with the immunity to tetanus, which remember is a bacterial toxin. and pertussis, again, a bacterial toxin, as it wanes over the course of our lifespan. And so these are things that if you're not chronically being exposed to, that immunity will wane over time. And so the recommended booster interval is every 10 years or during pregnancy. Likewise, we have COVID-19. It is as suggested, and this has a lot to do with the circulating types of COVID. And so I briefly mentioned the Alpha and Delta, and then the Omicron. And so this has to do with the new variants and also to counteract some of that waning immunity that we see about every 6 months. And then there is the measles, mumps, and rubella. And so this one requires a booster. Usually, this is a booster that's given in childhood, and that is required in order to properly stimulate the immune response to ensure lifelong immunity. And this is actually really important for adults born after 1957. And then we also have the rubella immunoglobulin G is tested during pregnancy because this is counterindicated. You don't want to come down with rubella during pregnancy and have a newborn with rubella because it can lead to some pretty catastrophic things. But if you find that you do not have immunity to rubella, that vaccine is administered as a booster after childbirth. So, fun fact, I was found to not be protected from rubella as an adult, despite having both in my series of MMR. And so I received a vaccine after the birth of my first. And then we have influenza or flu, and this is an annual, and this has a lot to do with the rapid mutation that happens with influenza. And so influenza 2025 to 2026, let's just review. We've got the process of antigenic drift or the inclusion of little mutations, and then we have antigenic shift where we have this recombination that's taking place to create a brand new virus. And so this could be a specific clade or even could be class switching. And so H3N2 is referred to as our seasonal flu. It's something that's been around for a really long time, whereas we have H1N1. Remember, these refer to the virus proteins that are expressed on the surface of the virus particle, and then H5N1, and that's our bird flu. And these all, the circulating forms when we switch from the prevalent type of H3N2 to H1N1, this is all through methods of antigenic shift. However, we also have this, the current influenza that is in circulation is H3N2 subclade K. And that occurred, the subclade K occurred through antigenic drift. And the vaccine that was developed using the H3N2 subclade J and J.2. And this was determined to be a mismatch. And this is a drift issue. So it still offers some protection against H3N2, but not high specificity to the subclade K. And like I had mentioned, H3N2 has been in circulation since 1968. But what ends up happening is in our manufacturing of the virus, scientists are using information that from circulating types of virus. And so what predominated the flu season in the Southern Hemisphere, remember that our flu seasons are flip-flopped, was H1N1. And it wasn't until the very end of the flu season that H3N2 clade or subclade K emerged. And so there wasn't enough time to develop an effective vaccine to that. And so when developing these influenza vaccines, the general considerations include the global surveillance of the active strains. And so we know that H1N1 is spreading globally, but H3N2, our seasonal flu, is predominantly subclade K. And then also there needs to be some prediction that takes place with the most likely antigenic drift, because those are things that scientists can actually monitor and make a best guess. So with that, that was a lot of information, but I really hope that the takeaway is that despite being small, viruses are incredibly complicated, and viruses behave completely differently depending on what they're made of and what cells they infect, and that vaccines can help prevent the spread, the unnecessary spread of different viruses. So, with that, I hope that I've got a question. Um, nope, I don't have any questions online. Does anyone have any questions that are, oh, let's see. Oh, we do have a question. Does a vaccine significantly reduce your chances of contracting said virus, or does it help reduce the potential severity of your symptoms, or both? So, this is actually a two-part question. So, I'm going to answer the first part, and then I'll ask the second question. So, having some vaccine or having some protection from a vaccine will reduce your chances of contracting a virus, and it will help reduce the potential severity because there is usually at least some crossover. And so, although with our vaccine that was released for the 2025 and 2026 season, although it is considered a mismatch, what has been found is that those that have been vaccinated have shown a much lower degree of hospitalization, which goes along that severity. And then it also does offer some protection from infection altogether. And so then the second question is also with viruses ever evolving to improve their own resilience, how often do vaccines get outdated and require a person to continuously get boosters? And so it really kind of depends on whether or not these vaccines have any type of seasonality. And so what we know is that with influenza in particular, we have these seasonal patterns of infection, right? And so right now, up in the Northern Hemisphere, we are right in the middle of influenza season. And so this is when influenza is spreading. It's spreading from person to person. It might have something to do with, being in close proximity to one another. It might be due to our overall dampened immune systems during the winter. It might have something to do with the sunshine because we do know that UV rays can help keep some viruses in check. And so viruses do evolve in such a way that they do require boosters, but we don't see that with some of the other viruses that have been managed well with viruses, including hepatitis B, MMR. So those aren't under evolutionary pressure because um, in regions where there are high vaccination rates of that, there isn't evolutionary pressure because the virus isn't actively spreading. And so the idea is if you can keep the virus from spreading altogether, you're reducing the likelihood or you're reducing evolutionary pressures for that virus to mutate and evolve and then have any type of viral or vaccine resistance. And so, yes, the general idea too with vaccines, or not with vaccines, with viruses, are to mutate in such a way that it becomes like the premier virus, right? The virus, for without trying to humanize it, a virus's objective is to infect. And so it will go through many, many different types of mutations, and those that show increases in the ability to infect other cells are the ones that are going to be naturally selected to spread. And so that process of infection is what drives virus mutation. And so if you think about people who have long COVID, so these are people that harbor COVID for 19 for long periods of time. Those are the people who have through no want of their own, right? It's not like a person chooses to be um, somebody with long COVID, but those are the people who end up having this virus go through many, many, many generations. And it's within those people that we have this evolutionary pressure for continual viral mutation and spread. And that is part of what drives this whole necessity for boosters if you want the protection, of course. And as long as we have people who are adamantly refusing to protect themselves through vaccine-mediated measures, we will have infection. And as long as we have active infection, we will continue to support viral evolution and the need for subsequent booster production. So great question. Thank you. Um, let's see. With that, I don't have any other questions. So I'm going to wrap it up. So if you do find that you do have any questions, by this time you'll have been watching on YouTube. So go ahead and put a comment down and I'll see if I can't find a proper way to answer it. So with that, this wraps up our Science Saturdays for January. There will not be Science Saturdays on January 31st. In February, we are going to be switching gears to plants and gardening. So I hope to see you in February. Thank you and take care.